Brad, you good? Okay, I think we'll get started while the folks are straggling in. I don't want to waste time for our speaker. So before I introduce tonight's Maury Strauss Distinguished Lecture, I'd like to put in a plug for some of the upcoming lectures. So next month we have Dr. George Benjamin, who's the Executive Director of the American Public Health Association, who will be here to talk about strengthening disaster resilience of academic research communities on March 28th. And that will be followed on April 18th by Sue Hockfield, who will talk about the age of living machines, convergence of biology and engineering, building the next technology revolution. Sue is the first female president of MIT and has a book out on that subject now that's garnering a lot of attention. And then in April, we have Marilyn Carroll, professor of psychiatry at the University of Minnesota, to talk about treating and preventing addiction. And then finally, to wrap up the series this year, on May 23rd, we'll have Steve Hyman. And Dr. Hyman is director of the Stanley Cancer Center for Psychiatry Research at the Broad Institute of MIT and Harvard. And we'll be here to close out the series towards the end of May. So I urge everybody to take a look at the website and stay alert. I'll try to send out the emails to remind you, but it's a great series. So before getting started, by reminder, this is the Maury Strauss Distinguished Public Lecture Series. I don't think Mr. Strauss is here this evening, but we're extremely grateful for his support of this series that allows it to be endowed and bring in these wonderful speakers at the Fralin Biomedical Research Institute. And we are pleased to have Haywood Fralin here, for whom the Institute is named. Haywood, thanks for being here this evening. And so without further ado, it is my great pleasure to introduce tonight's speaker, Dr. Kimryn Rathmell from Vanderbilt University. Dr. Rathmell is the Craig Professor of Medicine and the Director of the Division of Hematology and Oncology at Vanderbilt. She obtained her MD and Ph.D. degrees from Stanford University with her Ph.D. in biophysics that I just learned was not actually a medical school department, it was a separate department, biophysics, on the main campus. She then went on to do her residency in internal medicine and a fellowship in medical oncology at the University of Pennsylvania. After that time, she did postdoctoral training while at Penn, and then went to UNC, Chapel Hill, first as a postdoctoral fellow and then to join the faculty there. And she had faculty appointments in genetics, hematology, oncology, and became the Alexander Professor for Translational Science at UNC. And then just a few years ago, 3.5 or so, she moved to Nashville, to Vanderbilt, where she is the Craig Professor of Medicine and also Professor of Cancer Biology and Biochemistry, and as I said, directs the Division of Hematology and Oncology. Dr. Rathmell has won numerous awards for her research and many contributions. Just to mention a couple quickly, the V Foundation Award for Cancer Research, V Scholars Award, the Doris Duke Clinical Scientist Development Award, also the Outstanding Achievement Award of the World Congress on Cancer. She's published a number of articles, over 200 articles, and a number of chapters and books as well. And she was recently elected to be president of the American Society for Clinical Investigation. Her work started out in the early days studying DNA damage and repair of breaks in DNA strands, and did some very creative, interesting things then, and had an early interest, it appears from her publications, in renal cell cancers, particularly hereditary kidney cancers that she has carried on throughout her career. She determined early on the role of a molecule called a kinase, a protein kinase for DNA that's involved with detecting damage to DNA and triggering cellular responses. And for the scientists in the room, that's increasing P53 and modulating cell cycle and the cell cycle arrest. That's very important early work. She has gone on to do a lot of things with respect to renal cell cancers. And I just mentioned a couple things that I personally find particularly interesting. She's been studying the tumor microenvironment and the importance of human endogenous retroviruses that insert themselves through evolution into these tumors. And activation of viruses can, in some of these tumors anyway, can lead to an anti-tumor immune response. And that response can upregulate immune checkpoint pathways and then cause tumor sensitivity to immune checkpoint inhibition, for example. So this is a fascinating, relatively new area, I would say, that is opening up very important windows from fundamental understanding of the biology of tumors, leading to really some of the most exciting, in my opinion, some of the most exciting therapeutic interventions that we've ever seen. And this work is really on the front edge. I should add that Dr. Rathmell has collaborated a bit with one of our own faculty, John Chappell over here, when they were at UNC. I gather the labs that John was working in and Kimryn's lab got together and did some very interesting things on von Hippel-Lindau syndrome and have published several papers on that. So I know I've talked too much. I'm going to shut up now, and it's my pleasure to introduce, please join me in welcoming Dr. Kimryn Rathmell.
Wow, well, thank you so much for the kind introduction, and thank you for inviting me to be here today. I've had a wonderful day. I've really enjoyed talking to people and hearing about the diversity of work that's going on here. I was also really excited and a little intimidated when I heard that half of my audience wouldn't be graduate students and faculty members. So this is a little bit of a different talk to try to kind of bridge. It was fun to put together because it bridges most of the things that I do, at least. My clinical hat and my research hat. I am a researcher across the board, and that puts me in conflict with lots of companies because I do run a good number of clinical trials, and I have a spouse who's interested in starting companies and consulting. But I won't talk about anything that's relevant to that today. So I decided to try to put in perspective what I do and why I do it. And the problem is cancer, and this is a heat map that's age standardized mortality rate from cancer. This was from 2014, but was one of, actually it says 14 here, but I thought I pulled it from 2017. But why I work and live where I do is because cancer is prevalent here. Cancer is a problem everywhere, but as you can see, there's a strong signal right around Tennessee, and so this is why I work at Vanderbilt, is to try to make a difference in cancer in the bigger picture. You guys are in a nice little section that's yellow, so you're good. Okay, but why kidney cancer? So I work in the clinic, in the clinical trials, but really my research heart is in the basic sciences. And I fell into love with kidney cancer because there was real supply of questions. It's a very interesting cancer that violates a lot of the rules that get set up in other cancers. But most importantly, there's a strong connection between the genetics and cell biology and how we actually treat these patients. And at the time that I started my faculty career, which was in 2003, I'll show you a linear graph, we really had almost no therapies for this cancer. So you couldn't do anything worse. You could only make things better. And because it was coming of age at the time when there were more genomically driven or molecularly driven therapeutics, we introduced therapeutics that make sense rather than things that you just try and are lucky with. And so there's real-time changes occurring in patient survival. And I put this in italics because it's really true. In the course of my career, we changed from an outcome where every patient that I met, I said, you will die from your kidney cancer, to a place where I don't say that. This disease is now sometimes curable, and I'd love it to be most of the time curable. So it starts in the nephron. So this is what the inside part of the kidney looks like. So the kidney is a big filter and has a countercurrent multiplying system for sharing salts and gradients of glucose and oxygen. And so it's a really interesting environment for a cancer to occur in. Kidney cancers come from this outer cortex, this glucose high oxygen region of the kidney, and never come from this part. There are some subtypes of kidney cancer that arise down here, but the kind we're going to talk about today is all from the cortex. I will mention, though, that as I just said, there are many types of kidney cancer. This is my soapbox, is so that people know when you say kidney cancer, you should say which type, because it's really important to know the type. When I started my career, you just said kidney cancer, and you treated everybody the same. And in fact, until 2 or 3 years ago, you treated everybody the same. And the FDA indications, even today, treat them all the same. But that won't be the case for very much longer. So from the cortex, you get clear cell, and that's what we'll talk about today, the most common type, but also papillary type renal cell carcinoma. But from more of the distal tubules, you get chromophobe renal cell carcinoma, a disease called collecting duct, a very rare but interesting type called renal medullary carcinoma. So you can really split it up and study 10 or 15 different diseases when you say you're studying kidney cancer. So if I throw in just a few risk factors for kidney cancer, we don't have a lot. Most patients who have kidney cancer have no identifiable risk factor. We do have things like smoking on this list, but the increased risk from smoking is about 1.2 fold increase, so small. There is a clear genetic association with some kidney tumors, but these are strong hereditary conditions. I'll talk about one of them. Significant chemical exposures on the industrial level. Obesity, interestingly, is a risk factor and probably contributing to the rise in kidney cancer right now. So that brings to this slide. This is one of only a few cancers that is rising in incidence. We're seeing a lot of cancers that are declining, but that is not true for kidney cancer. I pulled UK data just because they had prettier graphs. But it's true there, as in the United States, that there's been over decades now a 2% per year rise in kidney cancer. It's most in developed countries, as far as we know, and it's rising a little bit faster for men than for women. In terms of the rising incidence, it's continued to project to be rising even in the rankings of tumors. It was considered number 8 in our big tumors list when I started. It's now number 6, and it's just going to keep going unless we figure this thing out. And it's rising in men and in women, but I put this graph up just to show that this is a male-dominated disease. So red and black are males. So it's white males, black males, and white females, black females. It's a little bit faster growth in black males, but historically, going back as far as we have good data, this is a 2-to-1 male to female cancer. Also don't understand that very well. But this is my favorite slide of the whole talk, which is the treatment revolution that's happened for kidney cancer. So when I started in 2003, we had 2 therapies that were available to us. One was called high-dose IL-2. It was only given in specialty centers. You had to admit patients to the hospital. It had a 5% death rate associated with it. And most patients weren't eligible because of the toxicity of the therapy. It was actually never FDA approved, but it was widely used. And then interferon alpha, which is a sub-Q injection, was never shown to have any benefit, but was FDA approved, interestingly. So that was it, right? Nothing, like there was nothing else. There wasn't stuff you used off-label. There was nothing. So, and then there was a rapid series of events that happened. So we'll talk in just a second about the von Hippel-Lindau gene. That's the most commonly mutated gene in kidney cancer. And one of the big downstream effects of having a mutation in the von Hippel-Lindau gene is a change in the vasculature. And I'll show you some data, actually some data from John Chappell's lab that illustrates that really beautifully. And so it was this connection between gene, mutation, and a phenotypic effect that we could see, which was the angiogenic change in vasculature that led to the approval of a number of drugs, all of the ones that end with nib, target blood vessels. So they target either VEGF, sort of the secreted factor that makes blood vessels, or the receptors on the blood vessels that help them grow. So sorafenib was first, then rapidly after that, sunitinib, bevacizumab, pazopanib, axitinib, and cabozantinib. All of these are anti-angiogenics. A couple of other things came along, mTOR inhibitors, temsirolimus and everolimus. And all of these drugs are sort of mood stabilizers. They really, none of these will cure kidney cancer ever. They can control kidney cancer. They were a great benefit to me in taking care of patients, and you could sequentially use them, and so you could create the sense of a stable disease, but ultimately resistance always occurs. And so that was the case until just a couple of years ago when we started to have immunotherapy. So nivolumab was the first therapy that was approved for use in renal cell carcinoma as a single agent. And then just last year, nivolumab in combination with ipilimumab was approved. So these are immune therapies. The activity of these therapies is to kind of unleash the native immune system and allow T cells to see protein antigens as foreign and attack the cancer. It's a very crude tool, and it's amazing that it works, but it works, and it works sometimes really quite well. And what we're going to see this year is a combination of what's called TKI, that's all these NIBs. We're going to see a combination of the anti-angiogenics with the PD-1, PD-L1, that's what these nivolumab, ipilimumab, that one's a CTLA-4. But combinations of these are going to be approved next. And the reason these will be approved next is because we've gone from a 5% response rate, but some stability. Moving up to some of the second generations give you more like a 30% response rate, but mostly stability. Nivolumab was only a 20% response rate, but some of them went all the way away. We call this Ipi-Nivo. Nivolumab, ipilimumab, 40% response rate, about a 5 to 10% CR rate. We call it, that's a complete response rate. That means no evidence of disease after treatment with this. This moves up into the 60% range for response rate. We don't know the cure rate with this, but we know it's probably in the low teens. So that's huge, and my clinic is completely different because of this. Okay, so back to basics. All of this came because we learned about the biology of the cancer, and there's more to learn, and we can continue to do better. So the origins of understanding clear cell type renal cell carcinoma, or what we call kidney cancer, came from discoveries in a hereditary disease called VHL, or von Hippel-Lindau. So VHL disease is a hereditary disease. It's a familial disorder of high risk for kidney cancers and other tumors as well. So all these patients who have VHL disease carry a germline mutation. It carries forward to half of their children. It's an autosomal dominant disorder. Although, while it is carried forward, a significant proportion, like 20% of cases, are first in their family. But it's a multi-system cancer risk syndrome, and it can be diagnosed sometimes even later in age. So it gives rise to kidney cancers. So most patients over the course of their life will have hundreds of kidney cancers. So this is not your normal kidney cancer. This is hereditary risk kidney cancer. They also have benign, but you don't want anything growing in your cerebellum and spinal cord. So benign tumors called hemangioblastomas. There are retinal angiomas that occur in the eye, and tumors that occur in the adrenal gland called pheochromocytomas. There are a few other things highlighted here where there are cystic or much more benign conditions. But for a really important gene, it's a surprisingly restricted set of conditions. So I run a clinic for about 100 of these patients, and many of them are family members of each other. And you manage this by just kind of keeping on top of things all the time, making sure none of these kidney tumors are getting out of control, that you've got good surgical control of the spinal cord, and et cetera. We are at the place where we'll have therapy for these patients probably within a year or two. The first clinical trial just completed accrual. So how does one protein cause so much trouble when it's lost? So VHL, here we have down here, normally regulates a protein called HIF. So it binds together with this protein called HIF, and VHL normally VHL normally binds to HIF. It causes it to get a tag that says you should be destroyed, and it gets degraded and it's gone. So it's made at a very rapid level, very rapid rate, and it's degraded at a rapid rate, and so the level you normally measure is very low. But in the absence of VHL, then HIF is free to accumulate. It can rapidly accumulate to high levels. So if you were to just eliminate VHL or its ability to bind to HIF, you would have high levels of HIF within minutes. So this is normally, HIF is called hypoxia-inducible factor because this is how your cell responds to low oxygen. So if a cell is starved of oxygen, it wants to, within minutes, figure that thing out and do what? Turn on a bunch of genes. So it's a transcription factor. It turns on genes. This is DNA. And so it turns on genes. For example, they're involved in bringing in blood vessels, because if you're starved of oxygen, bring some blood vessels and you get some oxygen. A whole bunch of other things are turned on as well. It changes your metabolism so that cell can survive the hypoxic period. There are also genes involved in basic survival, migration, because you might want to move away. Interestingly, it also turns on a gene that tells the whole body to just make more blood. So those patients who carry in their germline a VHL mutation actually have very high blood levels, interestingly enough. So this is why it causes cancer. So oh, there we go, there's John. I wasn't quite, didn't mean to get there yet. So it causes this very angiogenic, metabolically unique set of tumors. Okay, we'll just go to John. So this is John Chappell. And why is that important? So if you change the structure of the vessels, then that is a very nurturing place for cancer cells, in particular kidney cancer cells, to want to grow. So while the mutation itself is telling the cell, grow, don't die, do some easier metabolism, and by the way, I'll give you a super rich blood supply. So it tends to be a slow-growing tumor, but kind of insidious. And so this is just a model of what the vessels might look like in a VHL mutant. I had the pleasure of doing a project with John when we were trying to understand how these mutations could impact the vasculature. And so this is an experiment with a mutation in VHL that was introduced into a mouse, and then looking at the mouse retinas. So this is a picture of the vessels in a mouse retina at the 5th day after birth. And normally, the vessels, the arterial vessels and the venous vessel, and if it's anything wrong, please feel to just chime in, okay? Have a nice organized pattern. He did all this, by the way, I can't do this. Have a nice organized pattern. So this is essentially wild type, and we'll maybe just skip over here in the interest of time. When you induce some loss of the gene, so we actually have a mutation and a loss of a gene, then you can see that there's too many vessels, very disorganized, particularly in the arterial space. And so this is just quantified here. So we also look a little bit further along. So this is 21 days after birth, and you get disordered arterialization. So here's what a normal leaflet of retina would look like. Again, grows out nice and ordered. Here's that main central arterial branch. And when we knock out VHL, skip over to the farthest one, massively disordered, and in particular with increased arterialization. So that's what's going on in these kidney tumors, at least as much as we can understand that, because it's just a model, but it really helps lend some insight into that. And in those patients, remember, they also get retinal angiomas, so they're already having retinal disease and blood vessel disordered growth in their brain and spinal cord. I'll just also remind you that VHL does other things, and so VHL loss affects cellular metabolism too, as well as the angiogenesis. So normally, again, VHL binds HIF, it kind of gets out of the way, and so metabolism is often driven by glucose uptake, conversion to pyruvate, then in the mitochondria, driving the TCA cycle and eventually electron transport. Glutamine can help feed that cycle forward, and that's textbook metabolism, which goes all wrong. Goes all wrong in VHL. So in the absence of VHL, HIF is on. It's a really potent transcription factor. When I said it turns on genes involved in metabolism, it's lots of genes involved in metabolism. This is just a short list of things that are upregulated. And what does that do? We put these in this pathway. It increases glucose uptake. It pushes the metabolites of glucose uptake toward lactic acid, does not allow them to get into the mitochondria to fuel the TCA cycle. So now the TCA cycle is running lost, so it doesn't have the intermediates it needs to run this way. And so instead, glutamine also is increased uptake, and it'll drive that cycle backwards. And so you can make some energy this way, but it is a very, very disordered metabolism. So that's our first step in tumorigenesis. So from back to our nephron, here in the proximal tubule, an epithelial cell loses 2 copies of the VHL gene, upregulates HIF, and leads to an angiogenesis dominant disease. This is a figure showing that alternate mutations will give a very different phenotype, and I'm just not going to go into that for the interest of time. So beyond VHL, there are just a handful of other mutations that are involved in clear cell renal cell carcinoma. I talked to the students today about the great diversity of heterogeneity, but there are several core mutations that occur across the gene. This is a graph that shows, if you think about each column is one patient's tumor. This is from a national project called the Cancer Genome Atlas, where samples from across the country were brought together and analyzed for gene mutations and copy number changes and other genetic mutations to create an atlas of cancer. And so in clear cell renal cell carcinoma, the most commonly mutated gene is VHL. So this is mutations in VHL. VHL is located on chromosome 3 at position P21, and you can see that that is deleted almost universally. But the next most commonly mutated genes are all chromatin modifiers. So these genes, PBRM1, SETD2, KDM5C, BAP1, these are all things that affect the way chromatin is organized. And so we have major projects in the lab that I mostly won't talk about today, but compared to the other types, clear cell, VHL driven, and then a bunch of chromatin. High angiogenesis, high glycolysis. So as I told the students, though, it is a heterogeneous disease. So these are skeleton maps of the mutations. If you were to subsample around a tumor, you would think that you would get the same answer if you sampled here and here, but you don't. If you sample all over, you can draw a little dendrogram that tells you the trunk mutations. You can see in each one of these tumors in the trunk is a VHL mutation. VHL, 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 VHL. So that's universal. You're going to lose VHL somewhere. Also 3P, that's where VHL is located. So it's usually one mutation in VHL and then loss of 3P. And then there might be some other truncal things like PBRM1. Everything else is diverse. Okay, so it's heterogeneous beyond that. So this is just a summary of clear cell renal cell carcinoma. So VHL loss is the core. Remember that is, I think, one of the most important things. It almost exclusively occurs with a large 3P deletion and in a cell type that's uniquely influenced by oxygen and nutrient levels, highly angiogenic with an altered metabolic program. And the phenotypes are influenced, and I didn't talk about this much, by the specific mutant defects in the protein. That was actually some of our work with John. So I have several projects in the lab. We study metabolism, we study epigenetics, but we started to study a little bit of immunology because of this revolution in how we treat patients with kidney cancer. So this is an example of what it means to say that we've introduced nivolumab, our first line, our first in-class immunotherapy for renal cell carcinoma. Previously, the drug that would be compared in this setting would be everolimus, an mTOR inhibitor. Nivolumab was compared. Oh, I lost my label. This is a survival curve. So this is patients over months and looking at their progression of their disease. And so there was improvement, but it doesn't seem as good as I just hyped it up to be, right? In cancer world, if you can see through them, we get excited. Okay, so the scientists in the room, this was significant, and we get excited here. But this is not why we get excited. This is why we get excited. So occasionally, and it's not reflected well on this graph, you get patients that look like this. This is a patient of mine, and his before tumors show great big kidney tumor. Here we're looking at, here's his liver, normal kidney, here's his backbone. So great big kidney tumor. This is the way kidney cancer is. It's great big tumors, okay, because they don't get picked up earlier. He also had a bunch of liver metastasis. Okay, this is usually the writing on the wall, right? So we treated him for about 1.5 years on nivolumab, and everything just kind of slowly shrank, and then, until we were like, hey, you know, I can't find anything in the liver anymore. And your kidney used to be so big that it was kind of unresectable, but you know, almost anybody could take that out now, right? That's the oncologist speaking, not a surgeon. Anyway, I took him to the surgeon and said, hey, what do you think? And they said, I bet I could get that out. Let's see what happens. And that was 2 years ago, okay? This guy has been off therapy. He is essentially cured from metastatic kidney cancer. And I probably have 20 patients like that. I probably see 500. So some respond well, some blow through this. And so how do we convert more people to have this story? That's what inspires us. And so this first segment of the research is called Everyone Must Do Immunology. So this is really driven in the lab by Katie Beckerman, who just recently became a faculty member with us. She did a postdoc with my husband, who's an immunologist, but on the focus of kidney cancer. She did a little bit hybrid. And an MD Ph.D. student, this is Brad Reinfeld. So this is also Brad Reinfeld. And the other thing about immunology, just to take home, is that Jim Allison won the Nobel Prize this year for the discovery of the molecules that led to these drugs, like nivolumab and ipilimumab. So this is Jim. He has a band, and for reasons I don't know, long before he joined the lab, Brad is his bass player. So he's a scientist by day and a bass player for a bunch of scientists at night. So kidney cancer is also very interesting because if we go back to this atlas of cancers and look at the signatures of expression for immune cells, so you can decipher from immune signatures if there are T cells, so CD8 is a signature for T cells, part of our immune system, and rank all those tumors in order of gene expression, clear cell renal cell carcinoma, that's our cancer, is the highest. So we have the highest infiltration of immune cells, and we are a responding tumor, but it's clear that that's not the end of the story, because most tumors look about the same, and yet some respond and some don't. This is just a picture. If we were to stain we stained one of our patients for CD8, so these are T cells that are littered throughout the tumor. So if you take out a tumor and look in there, there's cancer cells, that's most of these blue dots. There's T cells, there's going to be endothelial cells, vessels, other stromal cells, but there's a lot of immune cells in there. So we're going to look a little bit at the metabolism, and so one of the questions that we're trying to address is, in that cancer, who is really using all the glucose? I told you that the cancer cells that have lost VHL have this whole pathway of activation that they should be the ones hoarding the glucose, and based on some of the stuff I'll show you a little bit later, our initial assumption was that some of what was going on in there was a bunch of stealing of glucose by the cancer cell that was depriving the immune cells. And so we've started to take a look at that by taking out tumors and actually seeing who's doing what. So this is a PET scan image from a mouse. So a PET scan here looks at glucose uptake. We take radio-labeled glucose, inject it into a mouse, take a picture of where it goes to the tumor. Everybody knows that that's because cancer cells love sugar. We measure how many cells are present. So there's more tumor cells than there are T cells in the tumor. This is compared to the spleen. And then we measure which one of those cells got more glucose. And when we normalize this to the number of cells, in fact, the T cells are taking up more glucose than the tumor cells. So these are this is who's hoarding the sugar, if anything. Although you'll see later, I think they're not even taking up enough. So I think there is some internal competition going on, but the T cells are the ones who really want some sugar. So that's not a big surprise. It turns out this guy named Jeff Rathmell has been studying immune metabolism for about 15 years. And what he's shown over many years is that effector T cells are very dependent on their nutrient availability and their selection of which nutrients to use to define whether they're going to go on to become a memory T cell or a cytotoxic T cell. These are the ones we want to have active in cancer cells, and so we really would like them to use glucose and less so depend on glutamine. That's a very simplified version of his work. So we started working with him, and partly because we moved to Vanderbilt, to be completely honest. He was at Duke and I was at UNC, and so we raised children together, but we didn't do science together. And then we moved to Vanderbilt, where our labs were right next to each other, and our postdocs started talking to each other. And so we started saying, well, let's see what's going on inside the kidney. So we started taking kidneys. I should have more of a graphic of what we do. So every patient who has a nephrectomy at Vanderbilt, a piece of their tumor comes to our lab. We do a single cell suspension, and we look at the tumor cells, we look at the immune cells. This is a experiment using a technology called CyTOF, which can look at multiple proteins at once and put cells into sort of buckets of like. So this is some subsets of CD4 T cells here on the right and some subsets of CD8 T cells here on the left. And so a normal resting profile of T cells would be mostly CD8s kind of falling into this bucket based on their expression pattern, and most of the CD4s falling into this bucket. And if you stimulate them, so in vitro we can say, go, activate, and they shift, and now their expression is down here. So this is just not activated, activated. So just sort of a profile for that. And so we said, what is going on in the T cells in the cancer? And it turns out neither of those things was true. In fact, they sit in sort of a separate space, and they don't activate very well, which I'll show you in just a second. This is just the profile that makes up those buckets. So this is an example of how they activate. So here is a normal example of activation on the left. So CD8 control T cells. So when we stimulate them, these are stimulated with CD3. An example of stimulation is increasing CD25. So peak on the left and then shifting to the right means activated. Increasing CD71, activated. increasing cell size activated. But the cells that we took out of the tumor, not so much, right? So that's why he's there. This is just a little bit more example. Here's again, normally proliferation happens when you stimulate cells. So our control cells, they start to proliferate, just bump, right? So they go just like they should. If we take blood from those patients, so this is a renal cell carcinoma patient, peripheral blood, and look at their T cells, they stimulate just fine too. Proliferate. But from that same patient, now we take T cells out of the tumor, not so. And so again, our original hypothesis was the environment must be just deprived of glucose. These poor cells must be starving. And that's not true. So when we look at the glucose or lactate in the space between cells, this is Brad's project. We call it the Tumor Juice Project. And we look at available glucose in the extracellular space of the tumor compared to normal kidney from that same patient. There's actually more glucose present. There's a lot more lactate present, not surprisingly. And so we certainly can't say that they're starved. Could it be that there's too much toxin or it's sort of toxic waste product? Maybe, that's possible. Now, Jeff Rathmell has shown for a long time that T cells can downregulate the essential things that they need to actually use glucose. You can give them all the glucose they want, and sometimes if they don't have the right cell surface or if they don't have the right tools, they can't use it. So we looked at GLUT1, that's a glucose transporter, thinking, oh, it must be low, but no, it's actually higher. So the TILs are in the black bar. So actually they have a little bit higher transporter. They actually have a little bit higher level of hexokinase 2, that's a rate-limiting enzyme of glycolysis. They actually have quite a bit higher phospho-S6. So they can take up glucose if they wanted to, but they're not activating. So it's not that they're missing key tools to utilize these nutrients. So in fact, they're probably taking up some glucose, but they're not really doing it right. And so the downstream effect of not really doing your metabolism right is to usually mess up your mitochondria. And so when we look at these T cells, their mitochondria are quite shattered. So the left side is control T cells, and purple is just the nuclear content, and off to the side is you have a nice thick fused mitochondria in green. So this is what a normal mitochondria would look like. And then in our T cells, you can see they're small, they're fragmented in all of the samples that we look at. And if you go look at it fast, this is just a movie kind of going through to prove that it's not. It is looping now. When I tried it before, it wouldn't go. I thought you would just get one chance. Anyway, it's a nice thick mitochondria, and here these mitochondria are tiny and disrupted. We look at it by EM, they're really disrupted and small, so here's our control. This is what a nice mitochondria looks like here and here. These are all to scaled. But when we look at our T cells that come out of the kidney specimen, mitochondria are tiny little balls. So this is just fragmented and small round mitochondria. And the effect of having a small mitochondria is that they're actually hyperpolarized. So this is measuring the polarization state of these T cells. So here's the TIL from inside the tumor is very high. So they also have very high levels of reactive oxygen species. This is the mitochondrial ROS is higher in the TILs compared to the control. But in the cytoplasm, reactive oxygen species is a little bit lower. And so I'm going to skip a lot of data, but to say, can we fix that? And we have taken several proofs of principle to show that yes, in fact, you can overcome these metabolic defects, at least in the cells when we take them out of the kidney. The next challenge will be, can we change this when they're inside the tumor space? So I told you they have high levels of ROS, so you can add a ROS scavenger, MitoQ, and in fact, that will take these cells from, so gray is unstimulated, black bar is stimulated, but without anything additional. But if you add MitoQ, now they stimulate. Both CD25 and CD71 showing that. I'm not showing you that if we add in pyruvate, so we just bypass all of glycolysis and feed them pyruvate, that will also overcome this. We can also block glutamine, and that will also allow them to overcome and rely on glucose as they should. So just to summarize this piece of the talk, The T cell metabolism is reprogrammed, but it is still modifiable. They are not in a fixed and broken state. The glucose can be available, but they'll fail to activate. Mitochondria are abundant but are small and fragmented, and they need the nutrients to be able to function properly, and that can be restored. These are all tools. These are not things that are ready for patients. I don't think we can just give patients high levels of pyruvate or mitochondrial ROS, although we are treating patients with glutamine inhibition and seeing what happens, and that these metabolic defects contribute to poor T cell function. So that's a project ongoing, but another immunologic project I wanted to talk about was the one that was mentioned in the introduction, which is looking at these endogenous retroviruses. Again, Katie was instrumental in this project as well, and Aguirre de Cubas is a senior postdoctoral fellow in the lab. So this is shifting gears a lot. So again, as was mentioned in the introduction, our genome contains a large number of endogenous retroviruses. In fact, if you look at this pie chart of all the stuff that's in our genome, the genes that have known physiologic function make up 3% of the whole genome. And then we always kind of think about, and then there's the junk, right? But the junk has a lot of other stuff in it. 8% of it is these endogenous retroviruses, which are, I'll go into in a minute, but they're ancient viruses that encoded and integrated into our genome. And other elements that are retrotransposons actually make up a huge component. So what are these viruses? So they're viruses just like other RNA viruses that we are infected with today, but ancestrally we were infected with RNA viruses that then were integrated into the genome. And there are hundreds and thousands of these all around the genome. I'm not going to go into all the details, but essentially, normally an RNA virus can be integrated, and then it can go be reactivated to make a double-stranded RNA and go on to be a virus and infect future other cells. So there's a core set of genes, and this is how we recognize these elements in the genome. And this is what sort of makes a functioning virus. And most of them are actually not functional. So that's because these are ancient, and over time, natural mutation has occurred and damaged individual components of these viruses. So mostly it's nonsense mutations. Those lead to stops or major deletions. And so most of them, you can look in the genome and find out that, oh, it's there, but it's probably not functional. But some can still be functional. So those that have an intact unit of LTR with GAG-PAL-N can have functionality, but they are typically deeply suppressed by histone and DNA methylation. So our genome has a way of shutting these things down. And so you normally don't see them. So when do they come on? So they come on with epigenetic deregulation of the genome. This is well known, and I'm going to show you some data where we're trying to take advantage of that. Sometimes in autoimmune diseases, they can come on and lead to the native immune system attacking host and seeing these things as foreign, which is, of course, what we're trying to accomplish with these immune therapies in kidney cancer. And some cancers, it had been previously reported in melanoma, Hodgkin's lymphoma, and colon cancer, and we reported it in renal cell. So why do we care? So why we care about these things is that they can elicit immune responses in a couple of different ways. One is that these retroviruses can get turned into RNA, and then be packaged as a peptide and presented as an antigen, just like any other antigen, and be considered as foreign. So we could potentially activate the innate immune system. Or just the fact of having some double-stranded DNA, a double-stranded RNA in the cell is not normal, and so the cell has ways of saying, alert, there's a virus here, we need to wake up, and that's the viral mimicry mechanism, which is a series of genes that are activated and mount some of the steps in an early immune response. These are called the interferon response genes. So let's step back. So kidney cancer, turns out, has lots of endogenous retroviruses expressed. I took out a couple of slides. We actually started by looking at all cancers, and there were only 4 human cancers that had normal, at baseline, had high-level expression of endogenous retroviruses. One of them was kidney. So this is back to that cancer genome atlas again. We keep going back and using that as a tool. So this is 500 or so kidney tumors, where we're measuring the expression of endogenous retroviruses. Red means high and blue means low. And so we have a set of lowly expressed, highly expressed, and intermediate. So when they're on, and when they're off. But we have a subset of tumors that have high-level expression of endogenous retroviruses. Interestingly enough, those that have high-level expression tend to do more poorly. So this is a Kaplan-Meier curve again. So patients who have low-level expression do better than those who have high-level expression. And we took this information, then went back to some of our own patients, and we had patients who had been treated with immunotherapy. We knew their outcome, and we took a look at their gene expression for endogenous retroviruses. We first looked at one called ERV3.2. It's an endogenous retrovirus that was, when it was high across different tumor types, so we thought it would be sort of a good go-to ERV. And those who had partial response had higher levels compared to those who had progressive disease. So that's PR and PD. Looking at two different probes for this. Or we could look at response rate. Those patients who had better response rate were those with positive expression of ERV. And if we look at progression-free survival, the patients who had high-level expression did better now. So they were patients who do worse if you do nothing, but they do better if you treat them with immunotherapy compared to those others. So these are probably those patients who have those good responses, and it makes that's sort of the difference in those curves, which look too similar between immunotherapy and non-immunotherapy. This is just showing another endogenous retrovirus. Like I showed you, there were thousands of ERVs that in mass were either on or off. This is just showing pink is responders. So looking across the components of this endogenous retrovirus, all the components were high, GAG, POL, M. This is one that we picked because it had an intact reading frame, and so it had potential to be actually expressed as a protein. And in fact, you can take then those protein sequences and find out if it is possible that within tumors that are responders, if they have T cells that can recognize those sequences. So that's that innate immune response that was on the right-hand side of the graph. Are they being presented as a neoantigen? Can they be detected? We don't know if it mounts a response. We just know that the answer is yes. It's easier to look at over here. These are two sequences from ERV 4700, and in those patients who were, the CD8 T cells of tumor were more likely to detect these sequences than T cells from healthy donors. So then questions emerge. Can we modulate ERV expression in clear cell renal cell carcinoma? Does ERV expression activate this antiviral response as well? And could we use a DNA methyltransferase inhibitor? So could we demethylate the genome to promote immunogenicity in response to immune checkpoint? I'm only going to show just a couple of pieces of data in this regard, but we used decitabine, which is a DNA methyltransferase inhibitor, and treated cells at a physiologic dose. Actually, we used two doses, so 100 and 300 nanomolar. And we looked at a cancer cell line and what's sort of a quasi-normal kidney cell line, and measured expression of endogenous retroviruses. And what you can see is from no treatment or mock treatment, 100 nanomolar, 300 nanomolar, we were able to induce endogenous retrovirus where it wasn't before. And does it mount the viral defense gene expression? Just put a couple of these up here. So it's just a reminder of what this looks like. These are the downstream genes, and when we measure them individually, blue is mock treated, 100 nanomolar, 300 nanomolar, they all are activated. So we are, in fact, just in cells and culture, able to increase viral defense genes. I can't tell you that this is coming from the ERV, but we're working on proving that now. So this is the current state of affairs. And I only showed you just a little data because I wanted to make sure we had time to talk. But that a subset of renal cell tumors, really roughly a third, express abundant endogenous retrovirus at baseline, that's associated with prognosis, which is poor, and immunologic response to immune therapy, which is good. That there's evidence for T cell receptor and engagement and viral mimicry responses, and that we can modify ERV expression with drugs that are used in some patients, and that we can also, at that same time, induce a tumor cell intrinsic viral defense signature. And I didn't show you, but you can that effect is sustained even after washout of drug. So that's the projects I wanted to talk about today. Were from the Rathmell labs. This is the village of Rathmell, which you can get to Giggleswick either via Wigglesworth or Wham. So this is to find your direction. It's in North Yorkshire. Can't account for anything. So finding the way VHL loss, HIF expression, angiogenesis, and tumor metabolism are the central dogma of clear cell renal cell carcinoma. And then endogenous retroviruses that are deregulated, we think because of the epigenome, can help to enable these tumors to be able to respond to immunotherapy and may serve as a potential source of neoantigen and something modifiable. And that dissecting the immune metabolic environment can also give us some new opportunities to intervene. And with that, I think I called out everyone. They're in bold here. If they participated, actually I cut out a section of the talk, so a couple people are bolded. They're important too, but we didn't talk about their work. We collaborate with a number of labs as well to get this all done. Acknowledge our funding, the Cancer Genome Atlas, which we were very lucky to be a part of. And this is our labs and my dogs. So thanks very much.
Kimryn, we have time for a couple questions. Yep, Jamie?
Is there anything, that's great, thank you. Is there anything specific about the epigenetic changes that lead to reactivation of the retroviruses?
Yeah, so not that we've been able to figure out so far. So there are all these epigenetic regulator genes that are mutated. We can't find a clear smoking gun that one of them is the cause. Way back when we didn't really understand this very well, like way back in 2010, we did FAIR-seq, this like ATAC-seq, but older, across human tumors to look if there were kind of changes to open and close chromatin. And there's clearly a signal of some tumors that have just massively open chromatin, but it's not clear to me what the cause for that is.
Specific to retroviral?
We have not been able to do that cross-validation between those two data sets. But in terms of, is it the SETD2 mutants? No, or the BAP1 mutants or the PBRM1 mutants, that clearly doesn't associate. So it's something else.
Question in the back?
From a layman's point of view,
Yeah.
we have two kidneys, correct?
Yes.
And when you find a problem with one kidney, if it's cancerous, how long is it before it shows in the other? If it does not, is a prophylactic approach taken to remove the bad one so that other one may survive? Does that give you better longevity?
Yeah, that's a great question. And actually at lunch, one of the graduate students asked that same question. So that's a great question. So detected early enough, removal of these tumors from the kidney is the way to go. 100% for sure. That's how you cure this cancer. It doesn't usually metastasize to the other kidney. If it's going to spread, it usually goes to lymph nodes, lung, bone, liver. So, but the problem is that it doesn't get detected early enough. We don't have a screening tool. There's not an imaging test. There's not a blood test. There's nothing that you normally can do to find this thing early. So the signs that a patient has that will indicate this usually don't come on until it's quite later stage. So blood in the urine has to be a pretty big tumor. That's why you get those big tumors. Pain in the flank. The kidney is not something that's really prone to pain. So these tumors are usually not painful until they're quite large. And then actually the other diagnostic criteria that we have is feeling that kidney in the abdomen, and it has to be really quite large for that. So most patients get picked up either incidentally, some blood in the urine, again, usually a pretty good sized tumor for that, or because it's already spread. So by all means, I would love to have an early diagnostic tool and turn this over to the surgeons.
Is that being worked on?
Oh, it is, but it's a little bit challenging. I talked to students today about another project that I have where we're trying to use contrast-enhanced ultrasound as a strategy to be able to detect early lesions with something that's not expensive, non-invasive. but it's not cost effective at this point to really move forward with that. It's not that, and the technology is really not quite ready for prime time. But or a blood test would be even better, but we're not there yet. Yeah.
Yeah, Francis.
So protocol hydroxylase inhibitors are clinical trials now to stabilize the bleeding to cure for anemia. Do you worry that you're going to have an increased risk for renal cancer with those?
Yeah, so yeah, I've talked to the people who are developing those drugs. So probably not, but I am a little bit worried. So it depends if these tumors are really just driven by pure HIF or if there's more. I think there's probably a little bit more to it than pure HIF. But if you have a pre-malignant lesion, adding some more HIF might not be so good for you. But what's really interesting too is there's another class of drugs. I alluded to this when I was on the VHL syndrome slide that targets HIF and inhibits HIF. So there's this new class of HIF2 inhibitors, and we're using them in kidney cancer, but I'm using them in VHL disease, and seeing this is really pretty, but really nice results so far, really quite impressive. But what's super interesting too, so I told you these patients have high levels of erythropoietin. I didn't say that, but that's how they get high levels of red cells. And so normally these people have a hemoglobin of like 15, 16, and I'm starting them on this drug and they're down to 9. So it takes them right down. It's really very interesting. So.
Front row here, question.
Basically, what I wanted to know is how fast can clear cell mutate? In other words, I know that you can come in with immunotherapy and it'll shrink a tumor, but also the cancer will mutate and defeat that particular.
Yeah. Yeah, so we talked about that at lunch today too. So there's a couple of things in that question. So one is how fast does this cancer even grow? It tends to be fairly indolent and grow slowly. So probably most cases are around for 5 or more years before they're detected. But what we're understanding now is it really depends on the order of events that a tumor gets. So back on that slide with all the little dendrograms. So they all get a VHL mutation to start, and they all get a loss of chromosome 3p. And then it depends what happens after that. So if they hit BAP1 and PBR1, that's essentially the jackpot, and those will grow fast. It's not clear if they'll be responsive to immunotherapy or not. We're still figuring that out. But if they get a SETD2 mutation, they grow slow, but with a lot of mutations. So they are hypermutable and really very interesting biology about how heterogeneous they are. And those ones probably will be able to find a way to evade whatever therapy we give them.
They're pretty sneaky, aren't they?
Yeah. So it's going to be a variable, though. We're going to have to know the biology of these tumors sooner or later. Right now, it is not standard to figure out what the genetics are of the tumor, but I would guess within just a few years we'll be wanting to know.
Ken?
With regards to reprogramming the CD8 T cells,
Yeah.
the caveat being that I know they're low CD25 expressors, can you add IL-2 to those cells in culture and reprogram them and kickstart those metabolic pathways as well?
Yeah, I wish Jeff was here. So they have done some of that. So you can get a better stimulation actually if you add CD28 as well, but I think you're asking sort of a little bit of a different question by adding IL-2. I'm not sure I can answer if they're able to completely reprogram. I don't think that they have looked at, so they'll activate better, but I don't know if we've looked at what they're doing in terms of glucose uptake.
So then my follow-up to that would be, have you looked at the T helper cells in the tumor? Do they have that exhausted phenotype, and are they low IL-2 producers?
I'm going to tell the postdoc you asked that because we ask her to look at that all the time.
Okay.
Yeah. So that hasn't been done.
I've heard one last question, Carissa.
Yeah, this is probably out of ignorance of where T cells come from, but you showed that the peripheral T cells are activating normally, so that would suggest that they're getting there, and then something is defective. Is there a way to kind of trace the origin of those T cells and see if that is the case where they're getting into the tumor, but in the normal tissue they're behaving fine and they can be activated normally in the normal tissue, but once they get in the tumor?
Yeah, that's a really good question, and one that's really hard. We talk about that a lot. What we'd rather look at is T cells that come out of the normal kidney. We've tried to do that. There's actually very few T cells in there. So we're not getting enough to really analyze, so that's why we went to blood. Our assumption is that they're circulating and then find their way there, but we have not been able to trace that and know that that's actually true. I also don't know if you take them out and put them back in if they'll come back that way. We need a good model for kidney cancer in mice. We don't have that right now. We're trying to create something like that so that we can do a little bit more of these tracer experiments.
Okay, please join me in thanking Dr. Rathmell. Excellent. That was terrific.
Oh, thank you.
So I get to give you a